I'm Vignesh Vashankar from West Virginia University, and I will be presenting you my work on the construction of a bioprinter from an open source 3D printer. The aim of this project is to create a bioprinter that is specialized in printing tissues with chemical probes in them. The probes will monitor parameters such as oxygen levels, pH, and phosphate concentration. This will serve as an alternative for live animal testing and for injection of probes into live animals. Instead, the probes will be printed into the tissue and will examine human disease tissue models without the need for animals. Building our own bioprinter maximizes flexibility in design and minimizes cost. So a bioprinter is a device which utilizes the 3D printing approach to create biological tissues and organs from a substrate called bioink. Bioink is a complex mixture of biopolymers such as proteins, cells, and additional compounds to support the cell's viability. Different types of bioinks and bioprinting strategies exist, and a few of them will be examined. The Ender 3 Pro extrusion 3D printer that is designed to print with plastic was remodeled. I replaced the plastic extruder, the stepper motor, the heated head, and the fan with a stepper actuator that connected to a syringe for paste extrusion. This setup was attached to an aluminum frame that rolled on the printer's conveyor belt. The syringe will contain the bioink and release it. I reprogrammed the firmware of the 3D printer using Ultimaker Cura to ensure operation with this new apparatus. The firmware updates adjusted the bed leveling and center positioning, turned off the bed heating, and calibrated the actuator's extrusions. A UV light source was added for photopolymerization, which I will bring up later. The image on the right shows how the 3D printer was transformed into its current form. To ensure that the attached syringe is capable of releasing bioink and at a proper force, a test was done with a substance with similar viscoelastic properties, hand cream. A few simple designs like WVU were made using the hand cream. In this process, a few coordinate referencing errors were identified and rectified. Now that the ability of the bioprinter to extrude a material resembling bioink was verified, a few bioprinting strategies were experimented with to see if the printer is capable of performing the tasks. 3 methods were tested: photopolymerization with resin, photopolymerization with gelatin methacryloyl, and fresh. The picture over here shows what varies among all the different bioprinting strategies. The first method tested was photopolymerization using resin, a synthetic material. Commercial resin with LINCBUO crystals is added to the syringe. The crystals are probes that sense oxygen and EPR images. This substance polymerizes in the presence of UV light. The spiral design in picture A was printed and produced the solid resin in picture B. The EPR image in picture C verified the presence of probes in the printed solid. This process, however, can only print out products containing the probe and not any cells. Alternatively, gelatin methacryloyl, a bioink, is used. This substance allows cells to be printed as well, along with the probe. The polymerization process is the same. It was verified that our printer produced successful prints with this bioink too. The last method tested is called FRESH, which stands for freeform reversible embedding of suspended hydrogels. This process involves a chemical reaction between the sodium alginate bioink and the calcium chloride in the bed. The cells and probes polymerize in the calcium alginate that is a result of the reaction. Since the bed was filled with gelatin, a semi-solid material, the needle sizes and speed of movement had to be adjusted. Here's a video of the 3D printer in action printing using the FRESH method. This is the final product after the printing is done. The polymerized product is suspended in the bath. The goals of this project moving forward are to perfect the needle movements in the gelatin substrate, ensuring a smooth print, to introduce live cells, and to perform EPR imaging confirming the presence of probes and live cells in the bioprinted material. On the technological side of this printer, my team is planning to introduce robotic arms that have multiple degrees of freedom. This will enable a more efficient method of printing. The significance of this work goes beyond printing biological tissues. This bioprinter will have the capability to print tissues with probes that monitor the chemical microenvironment. This is the first time that the chemical concentrations of bioprinted cells will be monitored. The long-term impact of this achievement will be the removal for the need of live animal testing. All kinds of drug testing and cancer research will be possible on bioprinted tissues. These are the works referenced. They provide information about the choice of materials and background on the printing strategies. You can also find them in the description if you're interested in reading them. I would like to thank the following people for working with me and playing a crucial role. Dr. Mark Saitlin is the primary investigator. Dr. Andrei Bobko supplied and designed the probes. Oksana Saitlin provided valuable advice on the structural aspects of the printer. Brian O'Connell, a PhD student and researcher, provided valuable biochemistry expertise. Duncan McGee prepared gels for the printing and conducted the trials. The National Institutes of Health, NIH, funds our facility. I appreciate your interest in viewing my presentation. If you have any questions or comments, please leave them in the comment section. Thank you for watching.